We talked about reproducibility and replication and how they're critically important statistical concepts in the medical literature. And we also talked about how the tools to achieve reproducibility and replicability already exist. So what's the big problem? It turns out the big problem is that humans are the people that are doing the data analysis. And humans are busy, they have different motivations, they might not make the data available or the code available in the way that you'd want. So an example of this is this editorial in the New England Journal of Medicine. It was written by the editors of the journal, and they came down in favor of people who generate data at the expense of people who maybe want to reanalyze it later. And they coined this term research parasites, people who take data that's already been published and reanalyze it and produce new papers. Now, certainly those people analyzing the data for a second time should be aware of the scientific and experimental background of the study. But making data available to a broader audience makes it possible to make new discoveries that we didn't make in the first time. This idea and attitude that data should be cloistered or kept in certain siloed groups is one of the reasons why reproducibility and replication are so challenging at this point in time in the medical literature. I'm going to give you a couple of little vignettes that just show why it's the humans and not the tools that are the problem. So here's an example of a paper that was published in JAMA, and it studied p-values in the biomedical literature. And this is an area that I'm excited about, and so I looked for the data at the end of the paper. And as with many medical papers, the data wasn't actually linked at the end of the paper. So I wrote the authors and I asked them, could you send me the p-values from the paper? And they responded kindly that they would like to do that, but it takes a lot of time to put the data up and it's so big, which totally makes sense. It's a complicated thing to do to share this kind of data. But the fact that the data weren't available was still a challenge and still a problem. Ultimately, they made the data available as a set of SQL dumps and they put it up on Dataverse. So this is great. They've made the data available, and it's a credit to them for doing that work. The data, though, were in a format that was a little bit hard to use. These SQL dumps are something that not everybody can use. But the cool thing about them making their data available is that I was able to write R code that would take those complicated files and produce something that's a little bit neater and a little bit tidier. So I could go back to these files, and I could find them, pull them down, and create an R package with tidy p-values that then can be shared and used by other people, citing the original work of the authors who did all the difficult work of collecting those p-values in the first place. So having that data available is now useful for other researchers who want to go back and analyze it. The point of this vignette is simply that it's a lot of work to produce reproducible code and data, and it's not always something that's funded or something that's supported in terms of the incentive structures in the medical field. And so that's one of the big barriers to making studies reproducible. Fortunately, there's tools out there now, including this one, on how to share data for collaboration that explain how to take advantage of these tools that already exist to make data available. This makes the process simpler, but it's still work, and it's still a lot of time that people have to spend on doing it. And it's so far not something that's entirely funded when you're looking at the funding for medical research studies. Here's another vignette focusing on replicability. Here I'm focusing on a study about fine particulate air pollution and hospital admissions for cardiovascular and respiratory diseases. This is a study where they're looking at the relationship between this air pollution and hospital admission rates. And they use a statistical model called a Poisson regression model. You don't have to know what that is, only that it's a very widely used statistical model where you can relate hospital admissions to a bunch of variables. For example, how much PM2.5 particulate matter is in the air, how many people are at risk, what day of the week it is and day of the year for cyclical effects, and what temperature it is outside, things like that. This statistical model is justified in many different books, but especially in this one, as being one of the most optimal approaches to modeling this type of data. The challenge is that while the statistical model itself is justified, there were a lot of decisions that went into how do we actually analyze that data. So they picked specifically 8 outcomes based on ICD-9 codes, 5 cardiovascular outcomes and 2 respiratory outcomes. They looked at hospitals related to injuries and external causes. They looked at only the years 1999 through 2002. And then the analysis was restricted to a specific set of US counties and those counties that had a high population. And it was also counties that were clustered into geographic regions. And the authors picked 7 geographic regions, but they could have picked other numbers. All of these decisions are human decisions. It's not clear whether they're right or wrong, but it does mean that if you wanted to replicate this analysis, you would have to replicate that decision-making process that the human made behind their statistical modeling. So when you're reading the sections about modeling data in the medical literature, keep in mind that many of the decisions that are being made are decisions that are based on gut instinct or historical data analysis done by that data analytic team. This idea is codified in something called the Garden of Forking Paths. Basically, the idea that when you get a new data set, you could analyze it in one of many different ways. And so how do you know that the way that you've gone is the way that everyone else would always go with that data analysis? And it's not always true that two data analytic teams will get the exact same answer. So how do we know if we can trust a paper if there's these potential issues like Garden of Forking Paths? Well, we can use pre-registration, things like randomized controlled trials. We already specify the analysis plan before we run the study, and that reduces the potential for this Garden of Forking Paths. If you see a study where they've conducted two analyses, where they've compared two different data sets and applied the same analysis and got the same answer, that would also increase credibility. Or where they've reduced the certainty of their statements, particularly in studies where they've only analyzed one data set or they haven't been able to pre-register, they might be a little less confident in the answers that they have. And then finally, if they use appropriate statistical techniques. And we'll talk more about that in the sections where we talk about statistical modeling. Altogether, though, the idea is that the tools exist, and as long as humans are committed to doing open, reproducible, and replicable science, it's possible to